Welcome to Build a Protein Workshop, Shaki.
It's been a while since I've seen you guys.
Well, you know us, we're always busy making molecules for the entire cell.
The day never ends.
How can we help you today?
Well, as a cytoskeleton, it's my job to maintain the structure of the cell. But lately I've been feeling a little bit weak. I need a new protein to help build my protein filaments.
Well, you're at the right place. I'm JC and I'm the ribosome, and I'm in charge of making proteins for the entire cell. I don't have a membrane, but I'm made of RNA molecules and proteins. Sometimes I'm free floating, and sometimes I'm found on the endoplasmic reticulum.
Well, as I said before, I'm looking to maintain the cytoskeleton, and to do that I could build a few things. So I was thinking I could build a microtubule, which is a strand of proteins that supports the cell and acts as a track. Or I could build a microfilament that also supports the cell and works with cell motility and the movement of cytoplasm.
Well, let's get started. So, since protein synthesis happens on the ribosome, I will use this RNA molecule to build a tubulin. So, let's get started. So, since
Hooray! My protein.
So, since the protein synthesis happens on the ribosome, my large and small subunit connect with the RNA strand in the middle, and then specific amino acids that correspond with the nucleotides form the protein.
Oh, and don't forget, the amino acids that form the protein they're told by the RNA.
Yes, that's very important. So let's get started. First, you must perform the heart ceremony.
Okie dokes.
Okay. Here's your protein.
Hooray! My protein! It's almost done!
Hi, I'm John, the vesicle whose phospholipid bilayer allows me to transport molecules throughout the cell. I'm here to help you move to the next part of the build a protein process. We need to modify and package your protein. Do you know that I'm also sometimes a lysosome, a vesicle that buds from the Golgi Apparatus and has digestive molecules that can break down things like carbohydrates, proteins, and phospholipids.
Wow, that sounds like a very difficult job.
But wait, I have more. Sometimes it can also be a peroxisome, which is similar to the lysosome, but it doesn't bud from the Golgi apparatus or have digestive enzymes. Instead, I'm mostly found in the kidney and liver, and I basically neutralize free radicals.
It sounds like a lot of work.
Yeah, we all do a lot of work, but it's okay.
Well, I believe it's my job because I package proteins and the lipids from the rough ER after the finished protein and lipid synthesis.
Oh, you're the Golgi body.
Yeah, I'm Chloe. So I will help to modify the proteins, and eventually I'll send them out through the help of John, the vesicle, and eventually they'll go to the cytoskeleton or other parts of the cell. Let me just modify the protein. It's gonna take a while. Having a little difficulty. Well, here's your protein.
Hooray! My protein! It's done! Okay, guys. Let's hit it.
Ready?
Build a protein at the Build a Protein Workshop, where there's a protein for every cell. Here's a protein, there's a protein, everywhere a protein. We're gonna have some fun. Ship it, label it, send it out. Package it, send it, synthesize it. Through the rough ER, synthesize the protein. Build a protein at the Build a Protein Workshop.
Proteins make up your cytoskeleton.
Thank you.